Students, today I am going to discuss about meiosis cell division. I am Dr. Mrigakhi Borah from Golaghat, Assam. Meiosis is a process where a single cell divides twice to produce 4 daughter cells containing half the original amount of genetic information. And meiosis occurs in germ cells of reproductive organs. For example, in males it occurs in testes, and for females it occurs in the ovaries. And in meiosis, as I already told, that one cell is divided to form 4 daughter cells. And meiosis cell division takes place in 2 phases. Meiosis-I, which is from one diploid cell, twice in number. This one is diploid cell, is first undergoes first meiosis-I division, and after that it is the mother cell is divided into 2 haploid daughter cells, and after this division these 2 cells again undergoes meiosis-II cell division to form 4 daughter cells with haploid number of chromosomes. The meiosis cell division is divided into 2 distinct phases. First one is meiosis-I, the second one is meiosis-II. And the meiosis-I is again divided into prophase-I, metaphase-I, anaphase-I, and telophase-I. And the prophase-I of meiosis-I is the longest stage in this cell division, and it takes place in 5 phases: leptotene, zygotene, pectine, diplotene, and diakinesis. And meiosis-II cell division is nothing but mitosis. It is very similar to mitosis, and it also takes place in 4 phases of prophase-II, metaphase-II, anaphase-II, and telophase-II. Now we will discuss about the prophase-I of meiosis-I cell division. Here the homologous chromosomes pair and exchange of DNA takes place between 2 non-sister chromatids to form recombinant chromosomes, that is, to form chromosomes with new characteristics. Here we should understand the meaning of homologous chromosome. A homologous chromosome is a pair of chromosomes which comes from each of the parents. That means one from mother and one from father. And these chromosomes have the same gene sequence, same locus, chromosomal length, and centromere location. Here is one pair of homologous chromosome, and these are the centromeres of these homologous chromosomes, and after replication it is divided into 2 chromatids. This is one chromosome. This is one chromosome, and this one is the other with two chromatids. The chromatids of one chromosome, one single chromosome, is known as sister chromatids, and the chromatids of two different chromosomes, that means this one and this one, or this one and this one, will be non-sister chromatids. Now we will discuss about Diplotene phase of prophase I, of Meiosis-I. The homologous chromosomes condense. The homologous chromosomes appear in the nucleus as in condensed form, and after that the centrioles divide. The centrioles divide in the cytoplasm, and in the next phase, which is zygotine, I will draw here. The homologous chromosomes become, this is the nuclear envelope. The homologous chromosomes, I will draw only one pair for my convenience. The homologous chromosomes comes closer. It will have two chromatids. Okay, this is two chromosome, that means one pair of homologous chromosome. And in this stage we will term them as tetrads. That means two nearby chromosomes of similar characteristics and similar shape and size, which we call as homologous chromosomes, comes closer, and now we will term them as tetrad. Means with four chromatids. And at this stage, this stage is known as synapsis. And when the chromosomes comes closer, a synaptonymal complex starts to form to begin in between the non-sister chromatids of homologous chromosomes to proceed for the next phase, pectine, where exchange of genes will take place. Next, this is pectine stage, and this stage the synaptonymal complex completes. That means the overlapping of the chromosomes completely takes place, and here this phase is very important, because at this phase the crossing over between the non-sister chromatids of each homologous chromosome pair will take place. I will fill up one chromosome to show you the difference. As at this phase, this is the this red chromosome which I filled is a different chromosome, and this one is the non-filled is the other. This is a homologous pair, and these two are, this one and this one are the non-sister chromatids, and these chromatids will overlap to interchange their genetic material. At this phase, the non-sister chromatids of homologous chromosomes overlaps and forms chiasma. This position is known as chiasmata. This cross-like structure, we will term it as chiasmata, overlapping, and crossing over takes place between two non-sister chromatids of the homologous chromosome at this place of chiasmata. Next, this is the process of crossing over. Here you can see that this is one chromosome and this is the other, and this is a homologous pair, and the interchange of genetic material or the crossing over takes place between these two chromatids. These two are non-sister chromatids, and interchange of genetic material, interchange of genetic material between these two non-sister chromatids will take place. The blue part comes from this chromosome, and this red part comes from this chromosome, and this is known as crossing over. Now the next phase is Diplotene. In Diplotene phase, the nuclear membrane starts disintegrating, and secondly, the synaptonymal complex degrades, and the homologous chromosomes becomes separated, but they remain attached at the place of chiasmata. The centromeres becomes apart, but the chromosomes remains attached at the place of chiasmata. Next phase is Diakinesis. The chiasmata and the nuclear membrane completely disappears, and the homologous chromosomes separate from each other. Now we will see the separated chromosomes like this. This part is interchanged part which came from the non-sister chromatid of the nearby homologous chromosome, and my drawing is not so good. Hope you all understand. Okay. Now these two parts are the interchanged part, or where the crossing over took place. These two parts. This is the overall picture of the stages of prophase-I. First, the condensation of the chromosomes takes place at the Diplotene stage, and second, at zygoting stage, the synapses between two homologous chromosomes occurs. That means they comes near. Third stage, which is very important, it is the Peckitin stage, where the crossing over takes place between two non-sister chromatids. Then third one is Diplotene, fourth one is Diplotene, and in Diplotene phase the synaptonymal complex starts disappearing, but the chromosomes becomes apart, but they remain attached at the places of chiasmata. And fifth one is Diakinesis. Here the chromosomes becomes apart and the homologous chromosomes becomes apart, and they will remain with their interchanged or new combination of gene sets. Now, after the prophase-I completion, metaphase-I stage starts. Here you can see that these are the meiotic spindle, or the spindle fibers, which are formed from the centrioles, which are placed at opposite poles, and spindle fibers remains attached with the kinetochores of each chromosome, and the homologous pair of chromosomes arrange at a double row. This is one pair and this is another homologous pair. Next, Anaphase I. At this phase, the meiotic spindle fibers shortens, and the homologous chromosomes move apart from each other. Next, the Telophase I. At this phase, the chromosomes become diffuse and the nuclear membrane reforms. The cellular cytoplasm divides to form two daughter cells of equal size with haploid number of chromosomes. Here we will find these two new cells with N or haploid chromosome numbers. Now the second phase is Meiosis-II, which I already told you that it is very similar to mitosis cell division, which has been discussed in my previous video. Okay, and it is Meiosis-II is composed of 4 phases. Prophase-II, where the chromosomes becomes distinct and condensed and spindle fibers form. Metaphase-II, where the spindle fibers attach to the chromosomes and the chromosomes align in cell center at the metaphase plate. And Anaphase-II, where the chromosomes separate and move to the opposite poles of the cell. And at Telophase-II, the chromosomes reach spindle pole ends and the nuclear membrane reforms. After that, lastly, the Cytokinesis-II, where the cellular cytoplasmic division takes place. That means the The cytoplasms of the daughter cells divide equally to form 4 new daughter cells with haploid number of chromosomes. Thank you.